Recording.
All right.
So when you're ready, you can feel free to share screen and take it away.
All right. Sounds good. Let's get this going. Let me just. All right. Like you said, I am. My name is Rory Eggleston, and I am excited to talk to you guys a little bit about the effects of stress hormone and stress on a certain period of vocal development in a wild species of parrot. Oh dear. There we go. Okay, so before we get into the nitty gritty, we need to do a little bit of background about what we're really talking about here. So vocal learning is what we're defining here as vocal production learning, which is the ability to modulate acoustic signals based on conspecific templates. So these are producing learned vocalizations. And this is a trait that is relatively rare. It's only present in a few vertebrate taxa, namely the passerine songbirds here, the hummingbirds, parrots, toothed whales, echolocating bats, pinnipeds, elephants, and of course us, humans. But this trait has been most well studied in the songbirds and humans. And through studying vocal learning, we know that there are, broadly speaking, are two developmental stages across vocal learning taxa. The sensory stage, which is when the organism, the baby, the juvenile, is kind of internalizing sounds. It's learning how to interpret those sounds and learning what they mean and memorizing them. And then they progress into a sensori-motor stage, which is when they are making the neuromuscular connections necessary to produce those sounds correctly and to produce them in the correct context. And so this transition between those two stages is very critical to the development of vocal learners, because it allows them to not only know the sounds that are being directed at them by other conspecifics, but also to produce those sounds and to produce them in the right ways and contexts. And part of this progression from the sensory to the sensori-motor is what's known as vocal babbling. And this is, again, a trait that's been found in relatively few taxa. It's known in the songbirds and humans most, but it's also been documented in parrots and echolocating bats. And broadly speaking, this is the exploration or incorporation of newer vocal variants and improvisation of new vocalizations into the vocal repertoire that occurs in a seemingly kind of random fashion. And this is something that occurs very early in development. In humans, of course, we know quite a bit about it. Babies tend to go through a well-known progression, depending on their parental language, of incorporating new sounds and syllables that progresses from sort of nonsense into definable words and then sentences. And something similar occurs with songbirds, and it's known as subsong. And in both of these taxa, in humans and songbirds, we know that it's a socially mediated behavior. That is, for example, in humans, babies will react to how their parents respond to their babbling and adjust the sounds that they're making accordingly. So if a sound, they don't hear their parent make a sound often or at all, then eventually they'll lose that sound. And only incorporate sounds that are used commonly in the language. So as I mentioned earlier, songbirds have been one of the main models that we've used for studying vocal learning. And we've learned quite a bit from this taxa. And an interesting thing to note, however, about the models that we've used are that most of the models songbirds studied to date only have song learning in males. And so this has sparked interest on how the endocrine system might interact with how vocal learning develops in songbirds specifically. So the endocrine system, the hormonal system, allows for internalization of environmental inputs. That is, when you're taking sensory information through your eyes or your ears or some other sensory modality, your body is able to respond to those inputs in some way, in a variety of ways that could be permanent or fleeting. And so we know that in the case of songbirds, or the models that have been studied at the very least, testosterone is crucial to song learning in male songbirds in these species. So testosterone basically activates the systems that allow for the males to learn song. And this has been demonstrated in a number of ways. Most notably, it's also been shown that if you give female songbirds testosterone, for example, they will also begin singing. And so this has spurred looking into other steroid hormones that are more sex neutral. And we found that other steroid hormones, such as stress hormones, can also affect vocal development, and again, are largely sex neutral. And this kind of leads to what's known as the organizational activational hypothesis, which posits that steroid hormones can permanently organize or modulate the nervous system during early development. And we can categorize these effects into two broad categories. Organizational effects, as described in the hypothesis, are effects that, when the hormone is present early in development, will permanently alter behaviors and physiology later on down the line in life, whereas activational effects will only occur when the hormone is present in the system. So now we're going to focus a little in on one of those sex-neutral hormones, corticosterone, or CORT. So we know that stressors are really important to development and can impact development in a number of ways. And when we're talking about birds specifically, there's a lot of stressors that occur in the nest, namely nutritional stressors. So if they're not getting enough food, nestlings will be impacted. Disease-related stressors, so things like avian malaria and other parasites. And social stressors, so competing with siblings for food, can all impact the development of nestlings. And when we're talking about stress in birds, that leads us to corticosterone, which is the primary stress hormone in birds. And briefly speaking, corticosterone is released when the hypothalamal pituitary adrenal axis, or the HPA or stress axis, is activated. So a stressor leads to the release of a cascade of hormones through the hypothalamus, the pituitary gland, and then the adrenal gland, which releases CORT, and then this seals a negative feedback loop that closes that cascade of hormones and allows the organism to respond to the stressor in some way. And CORT in birds has been found to depress nestling growth and feather development, but it can also have more positive effects, such as enhancing spatial memory. And again, these effects tend to be largely sex neutral. However, all of this is somewhat complicated by the fact that during early development, in some songbirds, the stress axis isn't actually fully developed yet until they leave the nest. So stressors that occur during that period of time when they're in the nest might not actually have any lasting or permanent effects. However, subsong tends to occur after fledging, so it's possible that it's a period of development that could be impacted by stressors, even in species where the stress axis doesn't develop fully until later on. So we've talked a bit about songbirds and how they've been used as a model, but parrots also represent a really good new model for vocal and social developmental research, and this is for a number of reasons. They've been found to be very plastic learners, so parrots are capable of learning new things throughout their lives. They're also extremely altricial, which means they're born completely helpless and take a relatively long period of time to develop fully. They also tend to have lifelong vocal learning present across the sexes, and they tend to have very strong pair bonds. And as you might notice, these are all traits that are very common to those of us, of humans. This makes parrots relatively good comparative models when we're looking at things like developmental behaviors and vocal development specifically. However, parrots, we don't know much about their vocal babbling stage and their endocrine system in any parrot species, and there's been very little development that studies that have been done on parrots, especially in the wild. But one exception is this species, the green-rumped parrotlet, or Phorpus passarinus. And a population of this species has been studied for over 30 years in Venezuela, and this has been facilitated by the presence of artificial nest tubes and the use of artificial nest tubes that allow the field teams to relatively easily access the nestlings, take video recordings, measurements, and closely follow the development of nestlings in real time in the nest. Moreover, the individuals in this population are uniquely marked and then tracked throughout their lives. And we do know a bit about their vocal development. We know that they have a learned signature contact call. So they learn contact calls from their parental templates, and these are used as unique signifiers. to identify themselves to mates later on in life or other conspecifics. So this species is a good option as a model to study the effects of stress on development in the nest in a wild system. So now just a little bit about what we're hoping to gain from all of this. We want to determine what the role stress reactivity has in vocal learning in this species. And our first hypothesis is that an acute response to social stress in the nest could foster the vocal babbling stage in this species via attentiveness, so how well they pay attention to their siblings, memorization of sounds from conspecific templates, and the development of their neural tissue. And some predictions that fall out from this hypothesis are that administering CORT could increase the output of babbling, the amount of babbling in individuals. It could increase the diversity of vocalizations produced in babbling. And it could also foster species-specific acoustical structural aspects of the babbling itself. However, a second hypothesis could be that the chronic stressors induced could impede vocal babbling in some way via developmental stress or the allostatic load, which is basically the wear and tear that chronic stress inflicts on a body. And some predictions that could fall out in this case would be either a decrease or elimination of babbling altogether from the development of these nestlings, a decrease in diversity of the babbling vocalizations, or inhibit in some way the reproduction of those species-specific acoustical structures. So now just a little bit about our methods. Again, the study site is in Venezuela. It's a ranch, Atomasaguaral. It consists of tropical savanna, gallery forest, and cattle pastures. And as I mentioned earlier, there are these 120 artificial nest boxes that these parrotlets regularly use for nesting. And you can see a mated pair here on the right. And in these boxes, you can rig video cameras. So the field team is able to get around 2 to 4 hours of video a day of the nestlings in nests that are currently under development. And so we can collect a really wide library and detailed library of development of these individuals. And as eggs are laid and nestlings hatch in these nests, the field team also can mark the eggs and nestlings with non-toxic felt tip markers. You can see the heads of these birds are uniquely marked, and this is for ease of identification for the field team and for people watching the videos. And when they get old enough, the nestlings get unique color bands that you can just vaguely see here. And these are used to identify them throughout their lives. And for this experiment, we used nestlings from 12 nests, and each of these nests have between 3 and 8 nestlings. And we used 12 nestlings for each of 3 treatments for a total of 36 nestlings. And those 3 treatments were either an 8 microliter dose of CORT mixed with vegetable oil that was given daily, 8 microliters of just pure vegetable oil to see whether the oil base had any effect or impact on their vocal development, and then a control of nothing. And the oil mixture and the oil CORT mixture were given via, delivered orally via a 25 microliter syringe. And this treatment period lasted for 1 week at around 21 to 27 days post-hatch, which is around the 3rd to 4th week that the nestlings are in the nest. So once the field team had collected all of these videos, we could analyze the video and audio in Adobe Audition. And we marked vocalizations as babbling if they fell under these identifying characteristics. If they were produced in the absence of parents, if they consisted of 2 or more structurally discrete vocalization types, and if those vocalizations were separated by 4 seconds or less of silence. Babbling also tends to be very low amplitude, so it's not very loud. And down here is, I had audio, but unfortunately my computer is on the fritz, so it's not working. But this is a visual representation of a bit of a babbling bout. It's a spectrogram. So a spectrogram is a measure of the change in audio frequency over time. So you've got kilohertz here on the y-axis and seconds on the bottom. And each of these little black areas represents one vocalization. So as you can see, there's a fair amount of variety in the vocalizations, and they have quite a broad range of frequencies, and they're occurring at kind of random intervals. So this is a good example of babbling in this species. So once we've collected those bouts, we were able to do a further acoustic analysis of the structures of the sounds in Raven Pro. And so I'm just going to go over a few of those measurements here using a spectrogram of a single vocalization. So you'll notice that there's kind of appears to be two mirrored things here. This vocalization is actually the sound at the bottom. This the second structure here is a harmonic. So that's basically something that naturally falls out from the production of this sound. But the sound itself is down here. So we had 8 spectrographic measurements, 4 measures of frequency: the upper frequency, the bottom frequency, the center frequency, and the bandwidth, which is the range of frequencies in the sound. Frequency modulation was another very important one, and that's represented here by this white overlay. So it's a measure of the average change in frequency over time. So it's an average of this increase, decrease, increase, and decrease of frequency, and how many, basically the relative change in slope of those frequencies. The inflection points of frequency modulation are every time it changes from increasing to decreasing. So there are 3 inflection points here. Entropy is a measure of the noisiness of the sound, so whether it's a pure tone or whether it's something a bit more messy, and duration is the length of the sound in seconds. And we only use babbling that was produced between 21 days post-hatch to 36 days post-hatch, which is basically until the nestlings fledge. So from the start of the CORT treatment until after. And a little bit about our stats. We had to log transform 6 of those 8 spectrographic measurements to normalize that data. And we use generalized linear mixed models to test for the differences between those 8 spectrographic measurements and between several other variables. And from these models, we used an Akaike information criteria score to rank the significant models and determine which model best explained the data. To estimate the repertoire size of the babbling vocalizations themselves, we used a k-means clustering analysis that utilized those 8 spectrographic measurements and then was plotted using a principal components analysis. And from those clusters, we calculated diversity using Shannon-Wiener diversity and Pialu's evenness. So now a bit on to our results. Across those 12 nests and 36 individuals, we collected 60,000 babbling elements. And on average, we found that nestlings babbled for around 6 days and produced 1,600 babbling vocalizations during that time period. And we found no significant differences between females and males in this study. Moving on towards the babbling repertoires and diversity of babbling vocalizations, that k-means clustering analysis converged on 28 clusters, which in this case translates to supposedly 28 different vocalization types. And again, this was mapped onto a principal components analysis, so the eigenvalues of those first 3 principal components was above 1, and therefore all 3 principal components were included in this analysis. And we found no differences between those 3 treatments, so the CORT treatment, the oil treatment, and the controls, for Shannon-Wiener diversity, H, or Pialu's evenness, J. But we did find a significant difference in Shannon-Wiener diversity between nests. So the diversity of babbling vocalizations differed between nests, but when you looked at the population as a whole, that difference fell out. So this is a visual representation of that cluster analysis, and I'll go through it a little bit. So each of these different colors and ovals represents one of those 28 clusters. So it should, in this case, represent one of those 28 different vocalization types. This ray biplot basically indicates how each of those 8 spectrographic variables explains the spread on one of the 3 axes that is included. Again, there are 3 principal components, so there is a 3rd axis in this cloud that's coming out of the screen towards you and away from you. And I'll go more into that in a bit. But basically what this says is, for example, duration and the inflection points explain the spread of this cloud along the y-axis, whereas those frequency measurements and entropy explain the spread along the x-axis, whereas frequency modulation, which as you can see has a short bit on that biplot, is because it's coming out of the It's more along the z-axis, so frequency modulation describes the spread along that third axis. And I've marked out 9 different vocalization types from 9 different clusters that you can see here along the edges. So something I just want to point out is that the vocalizations that are here in the bottom left tend to be very short. They are very short in duration, and they don't tend to have much frequency modulation. So they're just kind of flat. They're not increasing or decreasing in frequency. Whereas if you go over to the right side of the plot, the vocalizations begin to get longer, and they also tend to have more changes in frequency. So you have quite a few increases and decreases in frequency until you're getting to sounds that are basically just increasing in frequency and increasing very sharply in frequency. And as I mentioned, there's a spread along that third axis. And so this is just a representation is that the clusters are spread along 3 axes, and that's important to note here, is that the spread along that third axis is due to frequency modulation. So now a bit about our GLMMs. The only spectrographic measurement that we found was impacted by the CORT treatment was frequency modulation, which is again that average change in frequency over time. So as you can see, the average measure for frequency modulation was far lower for the CORT treatments than it was for the controls or the oil treatments. And this is interesting to note. This does not necessarily mean that the CORT-treated birds were modulating their frequency less. It just means that the average amount of their frequency modulation was closer to 0. So they could be, frequency modulation is a variable that contains negative and positive values. So when you're taking a mean of those values, you could get something near 0 if the organism is increasing in frequency a lot and decreasing in frequency a lot. Those values then cancel each other out. And once we found this, we were able to run several other models, including other factors, to see whether treatment alone was causing these effects. And we found that in the end, the model that included treatment and treatment period, which is a variable detailing whether the vocalization, a given vocalization was collected either during or produced during that week-long experimental treatment period or afterwards. And the interaction between those 2 variables, that was the model that had the highest weight. So briefly, what this means is that the effects of the CORT treatment lasted beyond the end of the CORT treatment itself. So once the parrotlets were not given any more CORT, this difference in frequency modulation persisted. And as I mentioned, this variable has 3 components basically for frequency modulation. An organism can decrease in frequency, which gives negative values. It can increase in frequency, which is a positive value, or it cannot change frequency at all and be flat, which is a neutral or 0. So while all 3 treatment groups tended to inflect, positively increase their frequency modulation more than decreasing, the CORT-treated birds had a more even amount of deflected and inflected sounds, which led to that lower overall average score. And again, this effect was seen to persist into the post-treatment period until nestlings fledged. So now to just kind of tie up all of that, I know I've been going on a little long. So what this means about CORT and its effect on frequency modulation. Because it impacted this frequency modulation variable and decreased the overall score due to both increasing and decreasing the frequency of their vocalizations in somewhat equal amounts. This suggests that the CORT is inhibiting the ability of these nestlings to produce more inflected sounds in some way, whether that be physiological or whether they're just not learning to do it more. And this is relevant because we know in this species that those learned contact calls that I mentioned earlier that are learned from parental templates also are incredibly inflected. So you see another spectrogram here, and this vocalization right here on the left, the second note, in case you can't see my arrow, it is something very close to a contact call. So contact calls have this sharp increase in frequency, and these are very biologically significant to this species. So if the CORT treatments, which are a proxy for stressors, are impacting the ability of these nestlings to produce primarily inflected sounds, then that could say something about how stress might impact their ability to learn species-specific formats in their vocalizations. And the other part of this that lends evidence that that stressors of some kind can in fact impact the biology and development of this species. We know that nestlings have a very robust stress response very early in development at around 13 days post-hatch, which is over a week before they even begin babbling. So this suggests that babbling could in fact be impacted by developmental stress in some way. And here we've shown that it's probably the ability to modulate their frequencies that is being affected. So a little bit more about their repertoires. We found that almost all of the individuals we used produced most of those 28 vocalization types. And we found that this babbling was cropping up at around the same time that they developed those learned contact calls, which again are learned from parental templates. So the fact that these are occurring simultaneously suggests that those 28 types might also be learned, much as babbling in humans and subsong in songbirds is also part of a learned behavior. We don't have direct evidence from this study, but it's a good start in the right direction to suggest that they are learned vocalizations. And finally, we found no significant differences in the output of babbling or the diversity of babbling vocalizations or acoustic structure between or across sexes. So dissimilarly to the songbird models that have been studied, sex steroids might not be playing a super large role in the development of vocalization, of learned vocalizations in parrots, whereas environmental stress might have very significant impact on the ability of nestlings to learn vocally. So just to tie all of that together, we found that the exogenous CORT, the CORT supplements, have had an inhibitory effect on acoustic structure, specifically frequency modulation in this species. And these effects lasted beyond the treatment period, which suggests that CORT might have organizational effects. So we might have permanently altered their ability to modulate acoustics and modulate their frequencies into adulthood. This study only monitored the birds until they fledged, but again, as they're uniquely marked, it would be relatively easy to track down these individuals as adults and see whether these differences have persisted into adulthood. We also found that vocal babbling is occurring concurrently with those learned contact calls, and therefore might also be a learned behavior, much like in humans and songbirds. And an important part of that is those signature contact calls, again, have evidence that their extreme inflection is a learned part of that behavior. So again, if the parrotlets are stressed can impact their ability to modulate their frequency positively, that might impact how they are able to produce biologically significant vocalizations like contact calls. And finally, we found no appreciable differences across sexes in this species, which suggests that more sex-neutral hormones like stress hormones can impact vocal learning in this species. And this is also interesting to note because songbirds, again, which tend to have song learning in males, are actually a sister taxa to parrots. So they're evolutionarily speaking very close together, but they have these kind of diverging methods of vocal learning. So this kind of lends credence to the idea that somewhere along the line, and part of that split is different modalities of vocal learning. Finally, I'd just like to thank everyone who's made this possible. My mentor, Dr. Berg, for all of his fieldwork and all of the access to this data. Soraya Delgado for all of her work, fieldwork and assistance with the audio-video analysis. My thesis committee for my master's, Dr. Klein and Dr. Mustard. Our amazing field team in Venezuela, who literally this would not have been happening if it wasn't for them. Norialbi, Mariana, Ricardo, Genesis, and Genesis. Dr. Mara Betancourt, for his design and interpretation of CORT manipulations, the undergraduate and lab assistant team who helped with the acoustical analysis, Leslie, Diana, Bridget, Logan, Clarissa, Natalia, and Brisa, and my lab mates, Caleb, Jessica, Abby, and Celia for all of their support. And this was supported by the NSF and conducted at Atomasak and UTRGV. Finally, sorry for going a little long. Any questions that I can take either here, or if you want me to put out my email, feel free to do that as well. I'll just keep this up in case people want to see slides.
A question from Sam. Do you want to unmute yourself, Sam, and come and ask it?
Yeah, I wrote it, so if you already saw it.
Yeah, just saw that. Actually, it was just the built-in one with PowerPoint. It has I think a relatively new accessibility feature that I found to be pretty good at translating my voice to text. So,
Thank you.
yeah.
I was really impressed with the accuracy of it, actually.
Thank you. Well, don't thank me. Thank PowerPoint.
Yes. I have a question. So you went into the differences between the parrotlets that were treated with CORT and those that weren't, with regards to the frequency modulation. But did you find significant differences on any of the other of the 8 axes that you were sort of dissecting the clips through?
Yeah, good question. We did not find any strongly significant. There were a couple of the frequency variables, so like center frequency or upper frequency, were edging towards being significant, but when we tested them further, it didn't seem to have any pattern or any effect really in this study anyway. So yeah, that's why we primarily focused on frequency modulation. Is we've tried pretty much everything else, and that's what's fallen out here.
Makes sense. Any more questions?
I'll just put my Twitter and email in or just my email rather.
Yeah.
In the comments if anyone wants to email
Fantastic.
questions later.
Thank you so much, Rory, for speaking today.
Thank you.
And thank you to Sam as well for giving your talk earlier. Hopefully Matthew will be back with us for tomorrow's seminars. Otherwise, I'm quite happy to come back and chair again. It's been a delight to have all you, and take care of yourselves.
Thank you.